we produce so much data that it is humanly almost not possible to find the correct data sets in these repositories. My name is Peter Schaap. I'm an associate professor at Wageningen University, and within the Unlock infrastructure, I'm responsible for the FAIR data platform. The FAIR data platform is designed to receive all the data that is produced within the experimental platforms. What it does, it makes sure that the data that is received is fair, that it is stored, and it also allows for analysis of this data. So what we did was to develop a simple tool, and the tool is especially for data producers, so you don't need a lot of knowledge to actually use the tool. There is not a steep learning curve because in the end it's all Excel based. And with this Excel sheet you can actually also on the fly almost verify your data. You take a sample, you immediately write down what's the metadata. And what the tool also allows is that once you're done or in between taking two samples, you can actually validate your data by simply uploading the Excel sheet to the data station. Well, I think there are three types of users. So one would be a person that doesn't think he actually needs the FAIR data platform. So that is an experimental user that actually only wants to use one or two of the experimental platforms. The second type of user would be a user that is also involved in doing omics experiments. Now, omics experiments are usually very large data files, and we have the capabilities to quickly analyze these omics data sets. For that purpose, we have actually developed a number of data analysis pipelines that by themselves are also fair. That is, they produce the data in a way that is reproducible and reusable. And what they also do is they also add to the metadata the parameters and the tools that have been used to come to this result. The third user would be someone who actually doesn't need to use the experimental platforms. For instance, if you take samples from the environment, you're not going to use these experimental platforms per se. But if they do omics analysis or other advanced analysis, the person can use these pipelines for further downstream analysis. And I think these are actually the three main classes of users that will use the FAIR data platform. Since your data is already verified with metadata, we have also developed a one-click upload system for submitting your omics data to the public databases. And that enormously helps you in getting the accession numbers required for publication purposes.